I'd now like to introduce our next laureate, Sir David Klenerman. He's the Royal Society GSK Professor of Molecular Medicine at the Yusuf Hamid Department of Chemistry at the University of Cambridge. A physical chemist, he completed his doctorate at Cambridge University with Professor Ian Smith on infrared chemiluminescence in 1985. Did postdoc work on high overturned chemistry with Professor Dick Zare at Stanford. And after 7 years with BP Research in their UK laser spectroscopy group, he returned to the University of Cambridge, and there his work was focused on the development and application of physical methods, particularly laser spectroscopy and single molecule fluorescence, as we're beginning to hear, to biological and biomedical problems. Recently, he's focused his work on studying the molecular basis of the adaptive immune response and detection and characterization of protein aggregates associated with neurodegenerative disease. Dr. Klenerman was knighted in 2018 for the development of high-speed DNA sequencing and also awarded the Royal Society's Royal Medal in 2018. Let me ask you all to welcome Dr. David Klenerman to the stage and present on single molecule fluorescence and its biomedical application. David.
Thank you very much. It's a real pleasure to be here today. So what I'm going to tell you is a little bit about my background, and then I'm going to explain to you a little bit more about single molecule fluorescence that we've already heard about this morning. Then I'm going to talk about DNA sequencing, largely focusing on its application, since you've already heard from Shankar the key ideas behind the technology. And then in the time left to me, I'm going to talk about how we're also using the same methods to study protein aggregation and neurodegeneration. And then one of the key themes I want to emphasize in the talk is how physical chemists build new instruments that enable us to measure new things and how that can enable new science. So this is me when I got my PhD, my undergraduate degree. So I wanted to study chemistry. I found biology too qualitative at the time and physics too abstract, so I focused on chemistry. Then I did a PhD. So this is the instrument that I built. So there's all this gas handling that I made myself. There's a home-built infrared spectrometer, and there's this big reaction chamber here. And what we were studying was hydrogen atoms that we made by microwave discharge, interacting with ozone, and that produced OH radicals that were vibrationally excited, and we used the spectrometer to detect the infrared radiation that these molecules emitted. I then went to the States, where I worked with Dick Zare. So what I learned from Dick Zare, he had an infectious enthusiasm for doing science. He was like a kid in a candy store. He really just loved doing science. And he really wanted to empower his students to do the best science they could. And that's something I've tried to do in my group. So in Dick's group, I tried to selectively cleave bonds in an organic molecule using a laser, which wasn't that successful. It's a very good way of heating the molecule up, but not cleaving the bonds where you wanted them. And then I returned to BP. And so at BP, I learned how to do multidisciplinary research. I was a laser spectroscopist, but I worked on very applied problems like how do corrosion inhibitors work, which the company used in the Alaskan pipeline to stop them corroding. So what I did here is I had this large laser table. If you've never seen a laser lab, we're normally in the basement in the dark. Here's a laser, a green laser. There's a dye laser here that's tunable to change the wavelengths. This laser here amplified the power of these pulses. Then it's relayed down to the sample, which in this case was a piece of iron in salt water, which was being corroded. And we were trying to watch the corrosion inhibitors bind to the surface of the corroding metal and see how they work. And then after that, I returned to Cambridge in the mid-90s. And at the time, one of the most exciting things happening in chemistry, and certainly in physical chemistry, was the capability to see single molecules. And the way that was done is as follows. You have a laser pointer exactly like this. You can even use a laser pointer. And we have a dye molecule, which is fluorescent. So if I excite the dye molecule, it will fluoresce a yellow photon. And I can do this process again and again and again. So typically, a single fluorophore can produce 1 million to 10 million photons, which are easily detectable with modern imaging technology. So a single fluorophore produces 10 million photons, I can easily detect it. And if I attach my fluorophore to a molecule of interest, a nucleotide, for example, or something else, then I can detect that molecule. So you work at very low concentrations, typically picomolars, so only one molecule is present at a time. And as Shankar showed you, it's the ultimate in miniaturization, and that's the idea behind this massively parallel way of sequencing DNA. But at the time, we weren't interested in sequencing DNA. Like Shankar's told you, we were interested in watching biology in action. So this is, again, another laser lab. It's all very colorful, purple, Cambridge blue, green and red lasers relayed over to an inverted microscope. The microscope has an objective, and you might have learnt in your physics class that light at a certain angle undergoes total internal reflection. But a small amount of the light, there's a decaying field, an evanescent field that propagates a couple of 100 nanometers above the glass surface. So here we have a T cell, which you're going to hear more about later on in the symposium. And this is a live T cell, and we've got an antibody that binds to the T cell receptor, which is responsible for initiating the adaptive immune response. And the antibody is labeled with a fluorophore. And so what we can do is watch the T cell receptor diffusing around on the surface of this live T cell. So this is what it's going to show you here. So each of those spots is a single molecule, and by analyzing this data, we can ask how do the T cell receptors diffuse around on the cell surface, or they're monomeric and dimeric, and how do they reorganize when in the initial early events of the adaptive immune response. So like Shankar's told you, we came up with this idea of using this technology to do a very fundamental study of how DNA polymerase incorporates nucleotides as it copies DNA, using two postdocs, Colin Barnes and Mark Osborne. And at the time, like Shankar's told you, it cost $3 billion to sequence the first consensus human genome, and then $30 to $50 million to sequence in individuals. And there was a real need, interest in getting this down to around $1,000 a genome. So we went to the pub. There's a local pub, the Panton Arms, just very close to the chemistry department. And the pub is not the important bit. The important bit is we talked to each other and I learned more about DNA and DNA polymerases. Shankar learned more about single molecule fluorescence. And that's how we came up with the idea of Celexa sequencing. The alcohol maybe have helped a little bit, but I think it's mainly the talking to each other. So we came up. So we didn't know quite what to do when we came up with the idea, whether we should write research grants. But one of my colleagues, who was a master of Christ College at the time, Alan Munro, persuaded us that we were going to need increasing amounts of money to develop the technology. And hence we decided that we were going to form a company, and the company was called Celexa. So it's a play on words. We had single and mol, and Shankar thought any good company had to have X in it, like Xerox. So we came up with the idea of Celexa. And we were thinking of having these DNA hairpins, so this is where this structure comes from. So, and it turned out that Alan gave us very good advice. So we came up with the idea, like Shankar's shown you, in 1997. In the year 2000, that there's this fly talking to some lizard at a garden party saying, well, have you had your genome sequenced? So this was just when the first average sequence of the human genome was coming out. In 2009, like Shankar's told you, we published our first human genome of an individual published, which was generated using our technology. So that's a very rapid 12 years going from an idea literally on a piece of paper to something that works and is published in Nature. And we couldn't have done that without forming the company. So it's gone off, the speed of uptake is well beyond what we imagined when we first came up with the idea. So this is a plot of number of genome sequence. It's a log scale against time. So 2001, the first genome. In 2007, when we published our genome, you can see that it really takes off. And it's now gone up to about 30 million genomes that have been sequenced, about 90% using this Alexa technology. Similarly, the cost has come crashing down, so up from millions of dollars all the way down now to $200 per genome. Like Shankar mentioned, in the UK, there's a 100K genome where they're sequencing individuals and then following their health records over time to understand the genetic contribution to common disease. And probably the most striking thing for me is this study. It's being used for newborn babies. So this was a newborn baby born in Addenbrooke. So it's the baby, the mother, and the father. And this baby was unwell when it was born, but it was sequenced using the sequencing technology. They sequenced the baby, the mother, and the father. And they can identify a gene that had been mutated. And in this instance, there was a treatment for the baby, and so on the television they showed a very healthy baby and two very happy parents because they managed to cure the baby. And this has been commonly used, certainly in the UK. In about 50% of the cases, they can identify what's wrong with the baby and they can be treated. The technology was used widely in the pandemic to look at how the COVID virus mutated and to then start to design new vaccines. So this is a plot of time, 2020 to 2021. Again, a log scale of the number of COVID virus sequences that were taken, again, largely using this Alexa technology. And I just wanted to highlight another interesting application. So this is Savanti Parbo, who won the Nobel Prize in 2022. And he developed a way of taking very old DNA from bone samples. So here's the Neanderthal, and he took a bone sample and managed to extract the DNA and sequence it. And again, he also took a sample from a cave in southern Asia. And he was able to identify, sequence the Neanderthal DNA or the Denisovan DNA. And from that, we could learn about our ancestry. So we had a common ancestry, and then we separated. This is Homo sapiens, this is Neanderthal, this is Denisovans. And so what he found is about 2 to 4% of our genome comes from our Neanderthal ancestry. So that's just another use of the technology, learning about where we came from. So in the other part of my talk, I want to talk about what we're doing now with the same technology. So we're very interested in understanding the molecular basis of neurodegenerative disease. So in neurodegenerative disease, you get these extracellular plaques, these intracellular tau tangles, or you get these intracellular Lewy bodies made up of alpha-synuclein. But it's not these sort of microscopic aggregates that cause the disease. It's the much smaller aggregates that have to form on the way to forming these larger aggregates that are thought to be toxic. And so we've developed methods to characterize these toxic aggregates, which are present at very low concentrations. And so we really need the single molecule sensitivity to do this. So this is not a cartoon. This is actually from a paper we published in Cell, where we aggregated alpha-synuclein in the test tube and worked out the mechanism. So I'll just talk you through this briefly. We start with monomer here. It forms small dimers and trimers by addition of monomer. And then what we found, these so-called oligomers can undergo a structural conversion into a more fibrillar-like aggregate that then can grow by addition of monomer into the large fibrils that make up the Lewy bodies. And what we also showed is that these small fibrillar-like aggregates were much more toxic than these aggregates. And this structural conversion was actually very slow. It took 24 hours, giving the cell some time to get rid of these aggregates before they converted into more toxic form. So, and then we had a sort of eureka moment in our study of protein aggregation, which was experiment one of my students, Matthew Horrock, did. So what we did is take cerebrospinal fluid, so this is the fluid around a person's brain that you get from a lumbar puncture, and so we took the cerebrospinal fluid of a patient unfortunate enough to have Parkinson's disease and put it down on one of our sensitive microscopes, and so all we did was dilute it tenfold and add a dye, thioflavin T. So thioflavin T is non-planar in water and non-fluorescent, but it becomes planar and fluorescent because it can intercalate inside the aggregates, inside the beta sheet, and so it becomes highly fluorescent. And when we did this experiment, we saw these small spots, and each of these spots we think is a beta sheet containing aggregate. So we were very excited because now we could look at the real aggregates that form in humans, but we couldn't measure their size because they were blurred by diffraction, and we couldn't learn about their composition because thioflavin T binds to any aggregate that forms a beta sheet structure. So I just need to have a slight detour to explain to you the other advance in my field, which is how you can break the diffraction limit and do super resolution microscopy. So this is an image blurred by diffraction, but what I can do is organize that I just have a few molecules fluorescing at one time. Again, this individual molecule is blurred by diffraction, so it's a 200 nanometer spot. But I know it's a single molecule, so I can replace this blurry spot with this higher precision spot. So typically, I can localize the molecule to 20 nanometer precision. And then if I do this again and again and again, I can build up a super resolution picture of the object of interest. So we're going to use that to look at our aggregates. So this is how we did it. We have a pegylated surface, so this is essentially a very non-stick surface to any protein. We have neutravidin, we have an antibody, and then we're going to capture monomer and aggregates of alpha-synuclein. And then we're going to come in with another antibody that has a fluorophore. It's not going to bind to monomer because there's no epitope accessible, but it's going to bind to the aggregates. And then we're going to make this fluorophore blink, and then we're going to be able to image the aggregates. And this is what we see. So this is a 100 nanometer scale bar. You can see these small aggregates that we see in cerebrospinal fluid. I mean, for me, they were surprisingly large. They're sort of the same size as coronavirus, so they're 100 nanometers. And what we find is there are also aggregates present in blood, in serum. Again, these are a little bit larger. And then the other exciting thing that we found is there were clear differences between the aggregates in the patients unfortunate enough to have Parkinson's disease compared to the controls. So the patients with Parkinson's disease tended to have more and bigger aggregates. So we're very excited that this might be a way of diagnosing Parkinson's disease early, so you can intervene earlier, ideally before people even have symptoms of the disease. The other thing we found is these aggregates are very good at causing an inflammatory response, not by the adaptive immune response, but by the innate immune response. And so we built an instrument to study this. So what we have here is a sheet of light that is focused on the surface of a live cell, and then we have a fine nanopipette, which we're going to deliver aggregates to the cell surface. So the tip of this pipette is typically about 100 nanometers. So it's like the pipettes you use at school, but with a finer tip. We're going to shoot out aggregates onto the cell surface, and the innate immune response is going to start, and we're going to watch that process occur in real time on a live cell. So this is just to show you, using the pipette, we're very good at delivering molecules. So this is a real-time video just shooting a molecule, wheat germ agglutinin, that has a dye on it onto a glass surface. So all these spots are just under a micron. And so what we're now going to do is have a protein called MyD88, which is labeled with a protein that you get from a jellyfish, green fluorescent protein attached to it, so we can visualize it. And what happens is the aggregate binds to a protein called toll-like receptor 4, and then you form a mydosome that contains 6 MyD88. So initially you have you just see a uniform fluorescence of MyD88, but when we trigger when the aggregate's delivered to the surface, you see these spots form. So maybe we can start the video again, okay. And so you can see in real time these midosomes form on the cell surface, and this initiates a whole bunch of biochemistry that leads to ultimately to the production of pro-inflammatory cytokines. So we're doing a lot of effort to understand how the aggregates cause triggering of the innate immune response, which aggregates are most potent, and understanding if we can block this process as a potential cure for Alzheimer's and Parkinson's disease. And I'd just like to finish, I think I have very similar conclusions to Shankar, which is probably a good thing. The idea came from basic curiosity-driven multidisciplinary science. It was a big team effort, and we really need to acknowledge all the people who worked in our labs at Selex and Illumina who made this possible. I think an important aspect of our success was that we talked and socialized as a multidisciplinary team, and I think this is where the best ideas come from if you want to do impactful science. I think you might only be at the right place at the right time once. So we didn't know if our idea was going to work or not, but we took the first steps of at least trying it out. The worst thing that could have happened is it might have failed. And as I've shown you, it actually worked well beyond our expectations. And again, like Shankar, my final conclusion is new chemistry, and particularly new ways to measure things which come from physical science, can solve major societal problems and improve people's lives. Thank you.